stupidity. Okay. Right. So yesterday I discussed, okay, of course, now Peaches comes in. All right, everyone's here. Everyone's ready to go. Let's do it. Yesterday, I talked about the structure of a chloroplast and how that is going to be related to the two stages of photosynthesis, which are the light reactions and the light independent reactions. Excuse me. I also talked about pigments, chlorophyll A, chlorophyll B, the accessory pigments to help the chloroplast absorb light. So today I'm going to go over the light reactions. All right, so the pigments that are involved in photosynthesis, which is chlorophyll A and the accessory pigments, which are chlorophyll B and the carotenoids, they're all grouped in clusters inside the membrane of the thylakoid. These clusters is called, well, each cluster, I should say, not these cluster. Each cluster is called a photosystem. We know of two types, photosystem 1 and photosystem 2. And the light reactions start when light hits those pigments. So down here, I've got a diagram of one thylakoid membrane and the electron transport chain. That's what this is called, electron transport chain. So you can see here we've got photosystem 2 and photosystem 1. Okay, so even though I can't describe both things at the same time, but just understand that light is going to hit photosystem 1 and 2 at the same time. And basically the same thing's going to happen after the light hits until you get to the end of each of those chains. So I have to describe one thing at a time. So starting with photosystem 2, light is going to hit the pigment and it's going to excite the electrons. The electrons, because they're excited, they're actually going to jump from the photosystem to something called the primary electron acceptor, or PEA. Okay, it is a protein that is in the membrane that is literally named for what it does. It is the first primary electron acceptor. Okay, so it accepts excited electrons from photosystem 2. The primary electron acceptor will then send the electrons through the electron transport chain, which is the next few proteins. And based on the diagram, you can see that if electrons leave photosystem 2, go to the primary electron acceptor, go down the electron transport chain, eventually they're going to end up at photosystem 1. So this is one important thing to kind of bookmark in your head or to highlight in your book, is that if photosystems lose electrons and those electrons are not replaced, eventually the photosystems are going to not have any more electrons left. So it is very important that the electrons that leave each photosystem are replaced. So if I ask you what replaces the electrons lost from photosystem 1, you say the electrons from photosystem 2. Okay, electrons from photosystem 2 leave PEA, ETC, they go to photosystem 1. So they replace the ones from photosystem 1. Now you might be asking yourself, well, what about the electrons from photosystem 2? How are they replaced? I'll get to that later, okay? Because I'm just going to go like in a row right now. So now we have photosystem 1. Now keep in mind, I did say at the same time that light hit photosystem 2, light also hits photosystem 1 and electrons leave. So by the time the electrons from photosystem 2 get here, those electrons have already left, right? So it's constantly being replaced. So the same thing happens for the electrons that leave photosystem 1. They go to a primary electron acceptor, they go through an electron transport chain, but here is where it's different. Okay, this is the big key. Because remember, the whole point of the light reactions is to take the light energy and convert it into chemical energy. That chemical energy is going to be in the form of two compounds, ATP and NADPH. That's where the chemical energy is going to be stored temporarily until we get to the Kelvin cycle, which is the light independent reactions. So the light energy gets transferred to the electrons, and the electrons are going to have that extra energy. They're going to move through these proteins, and eventually the protons at the end of photosystem 1 are going to combine. Now this is inside the thylakoid. This is outside the thylakoid. So at the end of photosystem 1, electrons are going to combine with NAD plus and a hydrogen ion to form NADPH. That's why you've got these two, what do you call it, an arrow that comes down, picks up the electrons and forms NADPH. This is 1/2 of where the chemical energy is stored. Okay, so remembering these two things happen at the same time. So light comes and hits both photosystem 2 and photosystem 1. The electrons from photosystem 2 and photosystem 1 become excited. They both leave and they both go to the primary electron acceptor, then get transferred down electron transport. So the difference between the two photosystems is the electrons that leave photosystem 2 are going to replace the electrons that left photosystem 1. And yes, they will eventually also get excited by light and then move on as well. But it's like you have to continue this process by feeding more electrons into the system, into the system. The difference is at the end of photosystem 1, instead of going on to yet another photosystem, the electrons here are going to leave the thylakoid. We're still in the stroma, right? That's important. We're still in the stroma, so we're still inside the chloroplast. And this is relating back to what I mentioned yesterday about how the structure of the chloroplast is so important to the functioning of the process of photosynthesis, because all of this is happening inside of a membrane, but it's inside the thylakoid, which is inside the chloroplast. So you're still within the same organelle, but you're able to have things move through a membrane outside of the structure inside the organelle, but with still staying inside the chloroplast. So once it gets here, then the electron combines with NAD plus and a hydrogen. And again, what I mentioned yesterday, you might be thinking, well, where did those come from? Where did NAD plus and hydrogen come from? They came from the second half of photosynthesis, which we haven't learned yet, which is why I said we learn the first half and then the second half, and then we're able to see where these things go. So the NADPH is going to go to the Kelvin cycle, which in turn is going to produce those two things, NAD plus and hydrogen. Okay, which then comes to the comes to electron transport, and then they're going to reproduce. So you see how it's a cycle, it's a continuing cycle. If these things are not cyclic in nature, then you're not going to be able to have photosynthesis continue for any length of time. Eventually, you're going to run out of electrons. If you run out of electrons, then the whole thing stops. You don't make NADPH, you don't go to the Kelvin cycle, you don't make organic compounds. All life on Earth is dead, right? So kind of important that you're able to, propagate or continue this process. In order to continue the process, you have to be able to recycle all of the compounds that are needed. So what we need is light energy. So we get that from the sun. What we need is electrons that are going to be replaced. So far, as I said, the electrons that leave photosystem 1 are replaced by these guys that leave photosystem 2. And in a second, I'll talk about what replaces electrons here. But at the end, we're producing chemical energy, right? We're changing light energy here and here, and we're storing it temporarily in chemical energy. That chemical energy, like I said up here, is going to go to the Kelvin cycle. That chemical energy is going to be used to drive the Kelvin cycle to make organic compounds, because that is big picture. Remember what I said about big picture? So if I look at the overall equation, and I should have started with this, so I apologize for that. I always want to keep in big picture. Yeah, that should be, oh, 2. Okay. Big picture, because we don't want to get lost in the details. The overall equation for photosynthesis is CO2 plus H2O yields, let's just use glucose as a representation of organic compounds, and oxygen. So far, we haven't seen where any of these, where any of the reactants are used. We also haven't seen where the products are produced yet. That's okay, we'll get there. But what I want to make sure to have you guys keep in mind is how all of these details are part of a bigger picture, like the puzzle that I talked about yesterday, right? Puzzles are not made of just one big piece, that's called a picture, right? A puzzle is made of one picture that is split into many different pieces. And so as you learn each little piece, you start to form all these connections that lead you to understand big picture. So this is big picture. Now I want to break down a little bit light reaction, Calvin cycle. So light reaction is going to produce NADPH and ATP. That's going to be used in the Calvin cycle. At the end of the Calvin cycle, oops, I'll just have it up there. At the end of the Calvin cycle, we're going to have organic compounds and oxygen. Okay, so that's the ultimate product. And in turn, in order to keep the light reactions going, the Calvin cycle is going to reproduce NADP plus and hydrogen, which we just saw in the previous slide. It's also going to reproduce ADP, and I'm going to call it PI for inorganic phosphate. Because how do you make ATP, right? I'll talk about that in a minute as well. So all of these things, I know it's like probably quite overwhelming to be like, oh my God, there's like so many things here. But what I really want to focus on is how all of this is connected to, guess what? Big picture, okay? So if this is the goal, food, right? C6H12O6, AKA food, that comes from this Calvin cycle. In order for the Calvin cycle to work, you need chemical energy, which is here. And then of course, light energy. Light energy. So the purpose of the light reactions is to take light energy. You have the light reactions, produce chemical energy. The purpose of the Calvin cycle is to take that chemical energy and turn it into organic compounds. Okay, move on that. Now, I mentioned before, photosystem 2 electrons are going to leave photosystem 2 and eventually replace the electrons that left photosystem 1. But how do you restore photosystem 2? Okay, that's where we have something called photolysis. Now, all that crap's gonna come back. Give me a second to erase all that. Really wish Keynote had an eraser option. Okay, keeping big picture up there. So photolysis, lysis to break, photo light. Okay. Now we have next to photosystem 2, we have something called the water splitting enzyme. And then here's where we see our first indication of why we need water. Okay, this is where it comes in. In order to replace the electrons lost from photosystem 2, we are going to split 2 water molecules. So we're going to first have 4 hydrogen ions. Next, we have 1 oxygen molecule, and then guess what? That's where this comes from. That's a byproduct, a waste product. Plants don't need it. And the last thing is going to be 4 electrons. Those electrons are what replace the electrons lost from photosystem 2. Okay, so now we see why we need water and why oxygen is produced. That's from the water splitting enzyme. Now these guys, the hydrogen ions, are going to be very important, when it comes to making ATP. and that is the last part of this puzzle. So far, we've seen how we make NADPH, and NAD, hello D, pH at the end of the electron transport chain, but we have not yet made ATP. But guess what? If we've got, let's say, here's photosystem 2, photosystem 1. This is going to be kind of messy. There's my electron transport. Okay. And then next to photosystem 2, I'm trying to put it all together for you. So I've got 4 hydrogens, 2 oxygen. This is photosystem 2, photosystem 1, and electrons. Okay. So it's kind of squished on the left-hand side, but I'm trying to put the whole thing together so that you see it all together. So you've got the same thing from before, photosystem 2, photosystem 1. Here's your primary electron acceptor with electron transport chain, blah, blah, blah. At the end of this, you have, let's see, what's a good color to use? This is black, I guess. NADP plus hydrogen. Whoop. No, too messy, sorry. Okay, hopefully that makes sense. Anyway, in the thylakoid membrane as well. So this is what I said before. The hydrogen ions are going to be really important in the production of ATP. So as you split more and more water to replace electrons lost from photosystem 2, you're going to have a buildup of hydrogen ions inside the thylakoid membrane. Again, this is where the structure of the chloroplast is so important. And again, there's a stroma outside, so you're still inside. Here's the chloroplast. You're still inside because this is one thylakoid. Remember, we've got like many stacks of thylakoids. So this whole system is repeated a lot in every single thylakoid membrane, in all of the thylakoids of all of the grana inside the chloroplast. You have an enzyme called ATP synthase. Synth means to make, ACE is an enzyme. So reading backwards or translating backwards, it's an enzyme that makes ATP. And it is driven, or it is powered by the diffusion, right? High concentration, low concentration. Because every time you split 2 water molecules, you are creating 4 more hydrogen ions. And you can imagine with all of the electrons that are being lost from photosystem 2, you're going to be splitting a lot of water molecules. So you're going to be creating a lot of hydrogen ions. You're going to have a very high concentration of hydrogen ions inside the thylakoid. They're going to diffuse through this enzyme, and that is going to drive the reactions that are going to combine ADP and a phosphate to make ATP. And now, based on the electron transport chain and the enzyme, you have the 2 molecules that contain the chemical energy that you captured from the sunlight. And these 2 molecules, NADPH and ATP, are going to move on to the Calvin cycle to help drive the Calvin cycle into making organic compounds. In exchange, as I said on the previous slide, oh, and I erased it all, of course, damn. You've got light reactions. I'll just do it again, it's no problem. And Calvin cycle. So you've got light comes in this way, and you're going to change the like I said, the purpose of the light reactions is to change light energy into chemical energy, NADPH and ATP. Those 2 molecules take the energy to the Calvin cycle, whose whole purpose is to make organic compounds. And in that process, it's going to make, when it uses NADPH, it's going to change to NADP plus and a hydrogen. Those are the 2 things that the light reactions need to remake NADPH. And it's also going to remake ADP and inorganic phosphate. So all of these things, again, is a cycle. The same way that I said all the way back in the beginning that aerobic respiration and photosynthesis are the same kind of thing. Photosynthesis produces organic compounds and oxygen. And those two compounds are used in respiration to have living things make energy. In exchange, respiration produces carbon dioxide and water, which are the requirements for photosynthesis. Again, big picture. There's, I think, no other two processes that I talk about in biology that are as closely related as this. The other thing that I want to mention, if I can get to a, let me get to a blank screen. Hold on a second. Okay. So we've got respiration, photosynthesis. So respiration produces CO2 and water. Photosynthesis, boy, crap, I just lost my Bluetooth connection. Lark, is it gonna stay? Nope. All right, try it again. Ooh, respiration, photosynthesis. What did I say? Oh, CO2 and water, the requirements. And then we've got C6H12O6 and oxygen. So that's how those two things are related, right? Now, if I go back to the overall equation for photosynthesis, CO2, we have CO2 plus H2O yields glucose plus oxygen. The overall for respiration is exactly the opposite. So again, it's how these two processes keep each other going. So that's why it's called a biochemical pathway. The products of one reaction are used as the reactants for the next reaction, and vice versa. I forgot, hold on, I'm gonna come back in a second. Wait a minute. Because I forgot to share the audio as well. Okay. Now we're inside the plant cell. Going into a chloroplast. There's the grana. And each of those flat pancakes you see is a thylakoid. And you see how all of the grana are connected. We're inside the stroma right now. Now, all of these little things with the rotating things on the bottom, that's ATP synthase. The green blobs that you see, those are the photosystems. And you can see what look like little flashes going between everything. Those are electrons. The little blue things are the primary electron acceptors. So you can see this whole system is just repeated everywhere inside the thylakoid membrane. Okay.